I'm going to present about the corneal fluorescence staining and my RNA sequencing experiment, which I did for my Pulner model. So this is kind of neuropathic corneal pen model. So the contents are the corneal fluorescence staining in neuropathic corneal pen models, some National Eye Institute grading system, how I analyze this staining, and the evaluation of gene expression through qRT-PCR in my future work. So first of all, what is corneal fluorescence staining? It's a kind of staining people are doing to evaluate the corneal health and to check the corneal abnormalities if they have any kind of epithelial damage. So it depends upon what kind of fluorescence dye you are using and how much the amount. So in my case, I use 0.1%, and I administered in my both pull model and normal control model. So for the normal control, I prepared the baseline image, and for pull neuro, I analyze it on the day 3. So what I found after that, there is some kind of analysis. So I searched and I found that there is a kind of National Eye Institute grading how people are analyzing the corneal fluorescence staining. So for grade 1, they have no 0 dots, and for grade 0, they have 0 dots, and grade 1 are less than 15 dots, and for grade 2 are less than 30 dots, and for grade 3 are less than or more than the 31 dots. So for analysis, we have to count the number of dots. So how we are counting the, like, first of all, this is my experimental data. This is the normal control model in on the baseline I prepared, and this is the uh, full nerve data on day 3 after my experiment. So if we can see, like, there's a no kind of observable damage to the ocular surface, and I I, I assess this on the stereoexome microscopic, and from the day 3, and what I found in the staining, like when I check it between the control model and the pull nano model, I found there is no significant difference between both these model. So that means there is no kind of epithelial damage after making this a model. So which is kind of significant uh, uh, significant for my model. If there's a kind of any damage to the epithelial or corneal, then the model will consider as some kind of disadvantage. So in my case, I didn't find any epithelial damage. So we can say that this is a kind of supported data for signifying our Pulner model. So after this, I'm also doing some kind of QRs RT-PCR. So we found some the top 20 upregulation genes. So they are the name of the genes. So we decided to go for SS3, KRT16, and LCN2 because these three are very important regarding to the corneal wound healing and the epithelial detachment. So in my case, in my HN staining, I found that the detachment of the corneal epithelial layer. So what I found, um, if we go through in detail, so I prepared something like in brief way, like what are the activities from the SA3 genes. So in a stromal cell, there is a kind of acute and inflammatory response, and some general suggested there is a kind of
Afzal,
if the gene,
could you make it like laser pointer?
okay, okay.
Yeah.
Okay, brother. So the second is about the neovascularization and alkaliber induced model. They told like if these are upregulated, then there's a kind of inflammatory response. And when there's a in another journal, they are saying like if these kind of activity are upregulated, that means both normal knowledge also have this kind of activity, and while the upregulation means there is a kind of inflammation goes on. And in the next for the KRT16 genes, what I found if these are the upregulated, that means there's a kind of cell addition and immigration, and this is a marker of activated proliferation in the epithelium. And also when the epithelial cell proliferate, there's a kind of keratinocytes migration, and this is also kind of role in the inflammation and wound healing. And in some cases, there is a overexpress in epidermis layer, which is kind of hyper proliferative area. So that means these genes are very much related with the epidermal layer. And the third one is LCN2 genes. So LCN2 is for the cell addition, migration, and autophagy information and post-translation modifications. They have diverse cellular process, including cell survival and death. And also, when we are considering inflammation, they have some kind of modulating immune cell function. So after this uh, brief summary and conclusion, we can say that the expression of SA3 in the cornea is correlated with the modulation of inflammatory responses and tissue repair process. And KRT16 is involved in keratinocyte migration and epithelial cell proliferation. And this is overexpressed in the hyperproliferative areas and bring corneal wound healing. And also LCN2 binding for the cell addition, migration, autophagy, and inflammation. So what I conclude, I want to go with the KR16 because these are the images from my pull nerve model. So we can see the epithelial layer of the cornea are damaged and it's a detachment. So what I found, according to the journal, if there's a kind of epithelial cell proliferation, that means the epithelium become eroded or detached. So I found the detachment in layer. That means my genes is my model is showing the activity of KR16 upregulation very high. How we consider? So we check with the qPCR. So I found the all three genes have showed the significant value, while the KRT6 have more significant relative MR expression. So that's why I want to go with the KRT16 because this is a kind of supported data for my epithelial detachment also. So that's it. And the future work is for about my other two model for the corneal fluorescein staining, which is related with the sham control and full translation model. And thank you everyone for listening. If you have any question.
Okay, yeah, thanks, Abjar.
Yeah.
Abjar, one thing I noticed is the eye fluorescence image, and that's probably it's a Taiwan YFP mice. That's probably why inside the pupil you see the fluorescence, green fluorescence, right?
Yes, Professor.
Okay.
This one is Taiwan white mice. So I'm just counting these dots 1, 2, 3, 4. So
yeah, potentially that may interfere if there are many inflammatory marks.
no, Professor. If it will only interfere if there is a very high inflammation. Rather, if there is a surface activity, corneal fluorescence is not undergoing below the cornea. So that means only corneal activity is showing, not the depth.
Okay. Yeah. Professor O, yes, do you have any comments or questions?
Yeah, Professor Zhang mentioned is it from Taiwan wife in mice,
Yeah.
right?
Yeah, Professor,
Meaning
yeah.
that this is the no fiber, right? Expression only, also expressed in other tissues in cornea.
Only the Taiwan is in the eye.
I mean the type of tissue, only fiber, no fiber, or other cells in.
This is the analysis only from the cornea.
No, sorry. The Taiwan YFP mouse, this YFP can be expressed in many parts of the nervous.
Cells.
Many.
Yeah, Professor. Yeah, it can explain the nerves and the axon also, both. Yeah.
Yes, we can only meaning the fiber, no fiber, right? No fiber.
Yeah. No
You
fiber
don't
also.
see anything.
Yeah.
Yes.
Mm.
But even you pull the nerve, you don't see any change. Even you induce some injury to the nerve.
There's some kind of pain in my behavior activity. The mechanical sensitivity, one flat test is increases, and the eye wiping test is also sensitivity, both chemical and mechanical are increased after the surgery.
But you don't see the fluorescence change at day 3.
Yeah, in corneal fluorescein staining, there's no kind of significant activity.
For your real-time qPCR study, how did you select these 3 genes?
For the 3 genes,
this is you screen from the sequencing.
This is the RNA sequencing.
Okay, I see.
So from these S100A8 and S100A9, why we didn't choose it, because they are very similar to the SA3. And
Okay.
GM1157 is very rare. It's only found
Mm.
in the only one rare disease. So I ignored this
Mm.
also. So then I found LCN2 is very high level in the retina. So I want to check if it's related in the cornea or not. And KRT16 is very much related with the epithelial deterioration. So
Mm.
that's why I choose for this one.
Okay.
So, like maybe for Afzal and maybe Professor O. So like these are coming from RNA sequencing data set, choosing these interested genes and confirmed with PCR that these genes are indeed overexpressed. But when I spoke with like other researchers who are asking whether this study, if these genes are very important, then it would be good to show that like knockdown or like overexpression, this overexpressed genes are knocked down, then is that really relevant to with a pain assessment so that it can provide a potential therapeutic genes? In that regards, I actually want to ask maybe Professor O, do you have any like suggestion or maybe not at this point, but maybe for future? Like, let's say, for example, here, LCN2, which is lipocalin-2, that's relevant with the inflammation and that's overexpressed. Then if we knock down that specific gene, which may be providing inflammatory like pain, then would that be like kind of interesting? Because then it may lead to somehow like not only we generate a mother, but also a potential for like key target for treatment.
Right. That's the always problem from this sequencing study, right? When you find some target from the sequencing data, you should validate. You already done from this real-time sequencing, right? The real-time RT-PCR, and then next question is what's the functional role of this overexpressed gene? Then to show that we need to knock down or you should manipulate the gene, then whether the manipulation changes the pain phenotype, then you can say this gene overexpression is associated with the pain phenotype. Yeah, that's the next step for this study.
Okay, yeah, I guess that makes sense. Like, so from that point of view, we may like kind of try to look for like which genes might be like critical for sustained change.
Yeah.
Right now, SAA3 is relevant inflammatory response. So if we, let's say, knock down SAA3, then maybe those inflammation might be reduced and maybe reducing pain. Potentially, would that kind of lead to somehow like more mechanism of our mother?
Right. Yeah, that should be the next step. Yeah.
Okay, I see.
But if you choose this gene, because if you can find some the other paper, right, like SAA3, how they involved in the pain, especially in the pain, that will be great, right? Because already somebody showed this gene is critical involved in the pain. Otherwise, you have to confirm, right, by demonstrating the pain phenotype. Yeah.
Right. At this stage, probably we will have to look for out of these three genes or maybe other, including a wider range of upregulated genes, and look for structure that like directly linked to the pain. Then we can at least discuss these potential targets for future study. Okay. Yeah, thank you. I think that's an important aspect. So maybe maybe Professor An or other remaining questions, if there's any.